In this module, we shall now learn about the methods for soil analysis and thus the comparison between different soil samples and also the different techniques used for paint analysis. Now, we'll first introduce ourselves to soil analysis. Now, basically, the question arises, why do we need soil analysis at all? Now, basically, what happens is that most of the time, mud and loads of dust, they are found on tires of the vehicles or on suspects' shoes. Now, although soil is quite different from other types of evidence, but still it serves the same purpose. So basically now our question is that whether the soil came from the crime scene or not. Now, although collecting soil sample from the shoes is easy, but the choice of our control sample is very difficult, which is actually due to the fact that the soil is a dynamic accumulation of particles which are constantly changing over very small distances also. Now, soil sometimes varies within inches across the landscape and also within inches down into the ground. Now, primary known samples, they are chosen from the spot where the crime has actually occurred. Now, a thorough analysis is then required to determine whether the two soil samples, that is the one which we obtain from the crime scene and the other which we obtain from either the tire of the vehicles or the suspect's shoes, whether they belong to the same origin or not. So, for this, methods like density gradient and observing some of the physical characteristics is very common. Now, coming to a slight introduction of what we are going to study in the paint analysis. Now, basically for the paint analysis, there are certain main concerns which are to be taken into consideration. That is the type of the case we are investigating, the sample size, what are the environmental effects, how much is the complexity of the situation, what is the condition of the paint at that time, and what are the collection methods that we are going to adopt. These factors actually lead the forensic scientists to the conclusion as to which method they are supposed to adopt for the paint analysis. Moreover, the paint films, they are characterized by certain physical as well as certain chemical properties. Now, the physical properties may include the color, the thickness, the surface, and the layer features, while the chemical properties include the pigments involved, the additives involved in the paint. Now, these features can actually be determined by various macroscopic, microscopic methods. Now, once the sample for the paint analysis is recognized, then it is removed manually with the help of various lifting methods and also by cutting. Then the analysis for the paint sample is done, taking into consideration that the method which we are going to adopt should be non-destructive and should require very less sample preparation. Now, these techniques include pyrolysis gas chromatography, scanning electron microscopic, and X-ray analysis, that is X-ray fluorescence and X-ray diffraction. Let us now come to a brief discussion about the different methods for soil analysis and the comparison of different soil samples. Now, in order to find a link between a suspect and the crime scene, the forensic investigators most of the time find it very easy as well as very beneficial to compare the soil samples itself. Now, for this, basically a very thorough analysis is required. Now, here basically we are going to discuss two very common methods for soil comparison. One of them is a very well-known method, which is that of density gradient method. Now, in this method, a cylinder is filled with various solutions, but in a very specific manner that the solution at the bottom will have the highest density, while the solution with lowest density will be at the top, and in between the density gradually decreases from bottom to top. Now, the soil sample that we have collected is then placed in the cylinder. Now the soil will keep on sinking through the solution which has got the higher density in comparison to the density of the soil, but the moment it reaches the place where its density matches with the density of the solution, at that time the soil particles, they will remain suspended and they will form bands in the solution. Now these bands, they represent the varying density of the particles which are making the soil. Now sample solutions from same origin will produce similar bands through this method. Now this is due to the reason that large deposits of soil, they have a uniform composition in regard to percentage of various particles, and also the density of each particle is always the same, as the density is an intensive property. This is a sample that the forensic investigators collect from the crime scene, and a sample which is found on the suspect's shoes or the vehicle's tires or suspect's clothing can be compared by this method. Now if both the samples, they have got the same origin, then they will show the same bands, but if they are showing different bands, then that means they do not belong to the same origin, that is, they have a different origin as shown. Let us now discuss the second method for soil analysis, which is nothing but a qualitative observation of the physical properties of the soil sample. Now, soil samples can be analyzed with the help of qualitative observations, which are based on sensory details and no mathematics is involved, which is again a very good thing in the analysis. In other words, the two samples can be prepared on the basis of their color, the texture, the particle shape and the mineral composition. Now the color of the sample can be viewed with human eye itself, but for that purpose the samples should be at the same moisture level, as moisture may change the color of the soil. Now for this, the scientists usually what they do is that they either dry the samples or they add extra moisture to both the samples so that their color can be compared accurately. Now the texture of the soil depends on the ratio of the sand, silt and clay, which are based on particle size and not on the chemical difference, which is again beneficial for the forensic investigators. Now regarding the shape, the soil particles, they have angular, sub-rounded or well-rounded shapes as shown in the figure. Now in order to examine on the basis of mineral composition, the forensic investigators, they use a microscope to determine the make. This is quite a reliable method as color and shape of a mineral is quite distinctive. Now if all these physical properties they match up, then they have the same origin. But if these properties do not match, then obviously they don't share a common origin. Now let us come to the next topic of different methods for the paint analysis and thus comparison of different paint samples. Now with the help of paint analysis, it is easier to actually identify the possible make, the model and in fact even the year of manufacture of the motor vehicles from the paint collected at the scene of crime or accident. It is also possible to differentiate a motor vehicle repaint from the original equipment manufacturer. Now once the evidence is recognized, now we try to remove it manually before tape lifts to collect evidence. Now it is collected with tape lifts very carefully, being aware of the difficulty encountered while manipulating paint samples bearing adhesive residues. Now if the contact between any two coated surfaces is indicated, then there is a possibility of cross transfers also. As a result, it is made sure that the samples from both the surfaces, they need to be collected. Now, paint flakes can be removed by a number of methods, like lifting loosely attached flakes, cutting samples of the entire paint layer structure using a clean knife or a blade, dislodging by gentle impacting the opposite side of the painted surface, etc. Now, once these formalities are complete, then for paint analysis, the sample size, the condition and the layer structure complexities is also taken into consideration while deciding as to which technique is to be adopted. Also, it should be taken care that more specific and less destructive tests should be done. Now, samples which are neither restricted by amount and nor by condition should be analyzed in such a manner that analysis will actually determine the color and texture of the paint, as well as the number, order, color and texture of the layers in case the sample is a multi-layered sample. Now, most of the time, instrumental techniques should be considered to analyze both the pigment, which is nothing but a finely ground organic or inorganic, insoluble and dispersed particle, and a binder, which is nothing but a non-volatile portion of the liquid coating of a vehicle which serves to bind the pigment together in the paint sample. Now, the first step in the paint analysis is the visual and the macroscopic evaluation, description and documentation of the original condition of the sample. Now, the next step comes into picture. Now, the next step of the initial evaluation should start with a critical review of the chain of custody, scaling of the package and identification markings of the sample. Now, if the package and identification markings of the sample are suitable for further evaluation, then a detailed accounting and description of the paint fragments should be documented. That is, in general, the size, the shape, the general condition, the exterior colors, etc., which is examined using stereo microscope, should be documented. Now, some of the techniques used for a detailed paint analysis are as follows. The first thing is the physical match. The physical match is the most conclusive type of examination that a forensic investigator can perform on paint samples. It involves the comparison of the edges and surface stray between samples. Now, both the edges and the stray in question must possess some unique characteristics. Now, when imaging methods are used to document a physical matching, then the forensic examiner should make sure that the method employed is dimensionally accurate and has got associated measuring scales. It should be made sure that the images are well preserved and retained as a part of the documentation. Now, then we have the microchemical test. Now, these tests are used to discriminate between paint films of different pigments and binder composition, which are already similar in visual as well as macroscopical appearance. Now, these are based on pigment and binder color reactions with oxidizing, dehydrating or reducing agents. Now, the problem with these tests is that they are destructive and should be applied to known samples first in order to know its efficiency. So, these tests should be used only in situation where the quantity of the question sample is adequate. Now, these tests should be applied to both the questioned and the known samples concurrently. The effect of these tests is then recorded immediately. Now, reactions such as softening, swelling, curling, color changes are some features which are to be noted down. Now, these results are then primarily used for preliminary classification and comparison. Then comes the polarized light microscopy. Now this technique is used for examination of layer structure and also for the comparison and identification of the particles present in a paint film, including the pigments, the additives, contaminants, etc. It involves the use of polarized light. The basis of this technique is nothing but the wave nature of light. Now, by polarized light, we mean the following. That is, a light wave consists of crests and troughs, which can be oriented either vertically or horizontally or any other plane. In other words, we can say that the light particles now vibrate in all the directions. Such a light wave is called an unpolarized light. Now, when this light is passed through a Polaroid filter or a polarizer, the polarizer blocks the vibrations either in vertical or in horizontal plane while permitting the passage of the remaining plane. Now, polarized light microscopy is based on the idea of polarized light. Now, in polarized microscope, there are two filters. One is the polarizer, while other is the analyzer. Light from the source move towards the polarizer and then is analyzed at different positions of the analyzer. That is, the two filters can be rotated in different positions depending on which spectrum will be formed. Now, if the two filters are rotated in exact opposition, that is perpendicular to each other, then no image will be seen. Thus, with the help of the second filter, different vibrational form of light will pass through the samples. Now, the different appearance of how light behaves on impinging through different samples of paint is made use of in determining the composition of the paint sample. Coming to the vibration spectroscopy method. Now, these methods we have already studied for the elemental analysis of glass also. Now, infrared and Raman spectroscopy, these procedures for which we have already discussed, are used to obtain information about the binders, pigments and additives which are used in the particle materials forming the paint. Now, infrared spectroscopy. Now, since the paint fragments are small, so Fourier transform infrared spectrometer is recommended. Now, out of transmittance or reflectance techniques, it is transmittance technique which is preferred, as most of the binder pigments, they have got emission spectra. In this type of spectroscopy, samples of different individual layers is prepared using scalpel, blades, forceps, etc. and placed on a salt plate or an appropriate mount. Now, coming to Raman spectroscopy, this technique is based on scattering of light rather than the absorption, unlike the infrared spectroscopy. So, in this case, the results are totally complementary to those that we get from infrared spectroscopy. Now, some paint samples, they may give rise to both infrared and Raman bands, but their intensity will be different in both the techniques. Now, coming to the pyrolysis gas chromatography, which is a very nice, a very safe, a very clean and a very common method. This is a destructive method of chemical analysis in which the questioned paint samples is heated to decomposition to smaller molecules, which are then separated by gas chromatography and detected using mass spectrometry. Here, pyrolysis means thermal decomposition of materials in inert gases or vacuum. The sample is put in direct contact with either a platinum wire or placed in a quartz tube and heated rapidly at almost about 600 degrees Celsius to 1000 degrees Celsius. The large molecules then break at the weakest points and produce smaller or more volatile fragments which are analyzed using mass spectrometry. Further, let us discuss a little bit about what do we mean by gas chromatography and mass spectrometry in general. Now, chromatography is nothing but a laboratory technique which is used for separation of mixtures. In this, the mixture is dissolved in a fluid called mobile phase, which carries it through a structure holding another material called the stationary phase. Now, the various constituents of the mixture travel at different speeds, causing them to separate. In case of gas chromatography, the mobile phase is nothing but a carrier gas, most of the time an inert gas such as helium or an unreactive gas such as nitrogen, while the stationary phase is the microscopic layer of the liquid or a polymer but on an inert solid support inside a piece of glass or metal tube called a column. The instrument used for such a technique is gas chromatograph. Now, mass spectrometry is nothing but a very famous and a powerful analytical techniques used to quantify known materials to identify unknown compounds within a sample. Now, the complete process involves the conversion of the sample into gaseous ions with or without fragmentation, which are further characterized by their mass to charge ratios and relative abundance. The applicability of this technique depends on the paint type, layer complexity, and the amount of sample consumption that can be tolerated. Coming to the scanning electron microscopy energy dispersive X-ray analysis. Now, this technique is used to characterize the morphology and the elemental composition of the paint samples. In this process, an electron beam, it impinges over a selected area of the sample, producing emission of various signals including X-rays, backscattered electrons or secondary electrons. Now, the emitted X-rays, they produce information regarding the presence of specific elements, while electron signals, they produce compositional and topographical visualization of the sample. Now, coming to the last techniques, X-ray fluorescence and X-ray diffraction techniques. Now, both are very famous non-destructive techniques. Now, because of the penetration depth of the primary X-rays, X-rays fluorescence analysis yield elemental data from several layers of a multi-layered paint fragments simultaneously. Now, this analysis can be used qualitatively as variations in layer thickness cause variations in X-ray ratios of elements present. While X-ray diffraction technique is used for the identification of the crystal form of the pigments and not suitable for organic pigments, this technique is based on the emission of characteristic X-rays after excitation of the sample by an X-ray source. Now, in the end, let us summarize what we have studied in this module. In this module, we have studied about the importance of the soil analysis and the paint analysis and what are the different precautions to be taken while collecting them and how to handle the soil and the paint samples. Now, as far as soil sample is concerned, we have studied that these samples, that is the mud and dust, can be found either on suspects' shoes or on vehicles' tires or on suspects' clothes. Then to determine whether the known sample and the questioned sample have the same origin, methods like density gradient and observation of physical characteristics of a sample are considered. Coming to the paint analysis, forensic investigators should take notice of the sample size, the environmental effect, etc. while doing the analysis. Now, paint films, they are characterized by various physical and chemical properties which include color, thickness, additive pigments, etc. Now, these can be determined by macroscopic, microscopic and instrumental methods like pyrolysis gas chromatography, vibrational spectroscopy and scanning electron microscopy.